So today we are going to discuss the topic when medicine fails, how antimicrobial resistance endangers wildlife. And today we are very, very privileged. We have two veterinary doctors that will be speaking to us on antimicrobial resistance. On my left, we have Dr. Jemima Mwangi. Dr. Jemima is from Kenya and works with IRI. And on my right, we have Professor Muso Munyeme. Professor Muso is from Zambia and is a lecturer at the University of Zambia. They both work on interface between the wildlife and human, trying to prevent things like antimicrobial resistance and zoonotic diseases. Dr. Jemima and Dr. Muso, we are very happy that you're with us today and you'll be speaking to us today. So for a start, we're going to start with Dr. Jemima Mwangi. Then she will do her presentation, and after that, we'll have Prof. Muso Munyeme.
Hi, good evening, good morning, and good afternoon. My name is Jemima. Thank you, Stella, for the wonderful introduction. I feel so delighted to see Professor Cooper and Margaret Cooper. I think I wanted to jump in and say, I know what I know because they really trained me. And
Wow.
We had a group, we used to call ourselves snake charmers. So what they're telling you are not stories. And it's a really big pleasure to see them here. Okay, I'll be turning off my video for bandwidth purposes, and then we can turn it on back again later. Okay.
Sure.
Thank you. I'm a graduate fellow. I work for the International Livestock Research Institute, and that is in short form ILRI, that Sela has introduced to us. And I wanted to talk to us about, or maybe in brief, about AMR in the world. AMR means antimicrobial resistance, and we want to understand what this threat is and probably see what the solutions have been, and come up, maybe know where to delve in partaking in this discussion. So thank you, Sela, for the introduction. For our talk this evening, I wanted us to have maybe a small outline. Let's discuss what AMR is, why should we care about it, how AMR in the world, because it's a discussion that should not be left behind, innovative solutions that have been, that are existing in AMR and what the researchers are doing, what can we do ourselves, and I would be delighted to discuss with you what I've been doing so far, and especially in the AMR field. So what is AMR? AMR is antimicrobial resistance in full, and what this when we use this term, we're trying to describe it's a situation where we find ourselves that microorganisms, that is bacteria, viruses, fungi, parasites, they have evolved to resist the medications that would, on a normal occasion, be effective against them. So you find either you have a bacteria, you're able to treat it with an antibiotic A, but now the bacteria has learned or has evolved to have a new mechanism that they are able to grow and thrive in the presence of that antibiotic that previously was able to either reduce the population or kill the bacteria in totality. AMR, I would say, is as old as before the invention of antimicrobials. We know that the first antibiotic was extracted in the 1940s from penicillium. But evidence have showed that AMR genes were present in ancient permafrost dating more than 30,000 years. So it predates human influence. It's not a new phenomenon. It's just that the natural evolutionary process that happens in bacteria in the wild or in their normal environment has been accelerated by our human activities. And some of these activities include misuse of antimicrobials, overuse. They accelerate its development and its spread. And AMR will develop. So this is a small description of AMR. So you have bacteria in your body, and then you take antibiotics. These antibiotics are supposed to kill the harmful bacteria in your body. So the blue bacteria here, they go away once you take the antibiotic, but a few still persist in the presence of the antibiotic. So what happens? Your body can either eliminate them or they still persist within you, but they're not causing infection. You can shed these bacteria, or they can multiply and later colonize you again and now give you another disease, maybe similar disease, another disease. And then when you take antibiotics during this time, because now what remained here and has multiplied are these resistant bacteria, then you have a new population that is resistant to the drug that you had taken before. You had pneumonia, you took antibiotics, it worked, some remained, they became resistant. The next time you take antibiotics, they're not going to work, or the action will be minimized. So that is what antimicrobial resistance is. I want to mention here that my bias will be on bacteria. I know microorganisms are many, viruses, fungi, but my work is in bacteria, so there will be that predilection towards bacteria. So that's what I'll be discussing, but the science is almost the same, how it develops, how to tackle it. It's different, but it's a basic understanding to what AMR is. So we ask ourselves, how does AMR develop? Genetic mutations in these microorganisms, for example, bacteria, they get random changes in their DNA, and this gives them a mutation against the antibiotic, which becomes an advantage for them. So the fight here is, you have a bacteria like E. coli, it multiplies. When you multiply in a family, what you get brothers, you get sisters, you get cousins, you have parents. Then we humans, we come in, we bring in antibiotics which kill E. coli's parents, E. coli's cousins. So what does E. coli do? If you fight my family, the general reaction is that I'm going to fight your family, to fight you. And that is what, to protect my family. And that's what antibiotics do. And that's what bacteria is trying to do, to try and defend itself from this thing that is killing it. So through changes in their DNA, they get mutations that give them ability to survive the exposure of any antibiotic. There's something called selection pressure from antimicrobial use. So we use a lot of antimicrobials, and we expose these bacteria to those antimicrobial drugs, and over time they become resistant, or the resistant ones are the ones that survive, like I've shown in this picture. They survive, they multiply, and then you end up with a population of very resistant bacteria, so making infection harder to treat. There's something else we call horizontal gene transfer. Unlike humans, microorganisms are able to share a few genetic elements, and you can share from one microorganism to the next, and that is what happens. Without going into details, it's two bacteria, they meet, they say, oh, I'm resistant, let me share this. We call it a plasmid. They share and they're able to pass on the resistance. So you end up with a population over time of resistant microorganisms. The key point is that with AMR, medications become ineffective, infections persist, and they spread. I'll give us an example how a bacteria is able to protect itself in a very simple way. So this is a bacteria. It has its outer membrane. I remember, if you've taken biology in class, bacteria is a living organism with a cell wall, with a nucleus, with all the parts that make it a living being, or a living organism rather. So it has an outer membrane, an inner membrane, and in between there's something we call a periplasm. So when you expose a bacteria to an antibiotic, the antibiotic gets inside the bacteria, or either it destroys, it has many mechanisms or many target sites for destroying this bacteria. It can either destroy the cell wall, leaving it naked. Of course, if your skin is taken away, you're going to die. It enters inside it. It has a target thing. Let's say if it's me, it's targeting my neck. If you cut off my neck, of course, I'm going to die. So that is what happens. That's how an antibiotic will kill a bacteria. Now, a bacteria has realized, they have realized this is what antibiotics do. So they try to come up with mechanisms to block the antibiotic. For example, here where my pointer is, you can see this is an antibiotic. This is its target, this ball here. So what will happen? The bacteria realizes if this is an antibiotic's target, if this one is killed, the bacteria will die. So what does it do? It comes up with a protection. So if this was my neck, what I will do is put on a neck guard, and then your sword won't go through my neck. So I'm protecting myself. Either another thing is a bacteria can have they form these small things, we call them efflux pumps. You give it a drug, it takes a drug, all right, but then the bacteria will invest in having these pumps to actively take out the drug. It comes in, the bacteria takes it out. You pump it in, it pumps it out. So those are some of the ways that a bacteria is able to protect itself without going into much details and too boring you. But these are some of the mechanisms of how a bacteria will protect itself against the antibiotic. So when that happens, what do we see? Now in the lab, what I do, for example, this is a picture. These ones, we call them plates. You grow bacteria on them. For example, in this plate, the white material, you can see the gray shaded material, that is bacteria that we've grown it in the lab in the appropriate conditions to allow it to thrive. Then we put antibiotic discs on the bacteria, on the plate, to be able to measure the effectiveness of a certain antibiotic against the bacteria. So somebody called John comes to the hospital, they have pneumonia, we take a sample, maybe sputum, we grow it in the lab until we have a pure isolate, we put it on this plate, then we subject it to antibiotics to see if John has this pneumonia, and in the hospital have this array of antibiotics, which antibiotic is best suited to treat John's pneumonia. So once in the lab, we have subjected the bacteria to these antibiotics. What you check for is the growth around the antibiotic, each antibiotics. Remember we said the gray area, those are bacteria. The white balls, circles, those are antibiotics. So if an antibiotic is to be effective, it's supposed to inhibit growth. So that, remember we said AMR is bacteria growing in the presence of antibiotics. So opposite of AMR is antibiotics being effective. So they're able to inhibit the growth, for example, here. You can see very well, these are, we call them zones of inhibition. We measure them to show effectivity of an antibiotic. So you can see on this plate, the antibiotics have been very, very effective against this bacteria that has been cultured in the lab. But on this other plate, however, the zone of inhibition is only large here. And I can say this is moderate resistance. And you can see in other antibiotics, there's totally no growth around, there's totally no inhibition, such that bacteria are literally having time of their life in the presence of that antibiotic. It's similar to having taken water. So that is what antibiotic resistance looks like. And why we should care is because this is going to cost us our lives, it's going to cost us our money, it's going to cost us our ecosystem. And we'll end up having existing diseases becoming more deadlier than they were, such that having a cat can literally kill you. That is what AMR means. We create superbugs, and we know that diseases, they have no respect for boundaries. Because of our migratory habits, you can have a disease in China and it's in Kenya the next day. So we should really care about antibiotic resistance, and the main drivers have been overuse and misuse. Of course, research is going on to prove this, to put more emphasis on this. So we should really care about AMR. And what is the scale of the problem globally? Annually, around in 2019, researchers were able to attribute 1.27 million deaths directly attributed to AMR, and that has been happening since then annually. These are deaths larger than HIV and malaria. I remember when I entered into the field, we used to call it a silent pandemic, but right now it's as loud as it can be. And other than these, another 4.9 million deaths have been associated with AMR, and these projections are showing by 2050 at least 10 million deaths will occur annually if we don't take any action, if we don't deal with this pandemic. And economically, cumulatively, around $100 trillion will go into this and will be lost through the burden of AMR. AMR in the world, AMR anywhere, I say for me is AMR everywhere. And wildlife are often victims of AMR and often victims of contamination because we don't rarely treat wildlife in the field. So if the driver is misuse and overuse, where would AMR be coming from in animals? Because this has been shown and I'll show us later. But the key drivers have been anthropogenic, either wildlife, for example, urban wildlife being exposed to human waste and food containing ARG genes, and microbials used on livestock and on the farm, and then those that persist in the water and in the soil. All this facilitate transmission of AMR genes into wildlife. Natural evolution has also been shown to occur. For example, penicillin-resistant Staphylococcus aureus was expressed in hedgehogs. So that is something that happened outside overuse and misuse. Wildlife really matter. I remember we said we really don't treat wildlife. So where are they getting AMR from? But other than that, wildlife, they serve as bioindicators of AMR pollution in the environment. They help you know what is the contamination level in an environment. They mirror what is in the environment. So if you wanted to come to Nairobi and test the level of contamination in Nairobi, I'll tell you, don't go sampling the rivers, don't go sampling the garbage sites. Look for rats, look for marabou stalks, look for the wild birds in the area. Sample those, they will tell you how Nairobi looks, or the area you want to sample looks like. They are victims and they will become the sink of livestock and humans because they do not use. It means they get it from somewhere else. The source in them has to be an external source. There's the theory of this, the source and sink theory, that says the more you have, the more the likelihood you are the source, and the less you have, the more the likelihood you are the sink. So if you find AMR in wildlife, you ask ourselves, are they victims or are they villains? And I've seen papers showing the difference between the two, either wildlife are the victims or are they villains of AMR. Wild animals possess a long range of movement, and that becomes an essential drive of AMR because of the risk of spread. For example, if you have a firefinch, or I don't know what birds you find in Europe, that there's a quite an ubiquitous use of, let's say, colicitin or meropenems in Europe that we barely use in the Masai Mara. And this bird picks it up from the environment. When it's winter, there comes to summer in Nairobi or down the Masai Mara because it happened and it carries these genes with them. So where are we going to say a buffalo in the Masai Mara got this gene from? So long range movement, it suggests that wildlife can become distant connectors and reservoirs of AMR. However, Africa is very rich in wildlife, yet data is very scarce, or studies have been very minimal, and I will show that. I was able to do a systematic literature review of wildlife studies conducted in Africa, in African wildlife, to showcase or to test for AMR, either in surveillance, in treatment, and we're able to extract 144 studies from only 21 countries in Africa, like I've shown in this map. Very few papers per country, only very few countries had large numbers of papers. The duration we went back to, this was not purposive. We opened the duration, but the oldest paper we were able to find was in 1980 of buffaloes that were tested in Kenya, skin bacteria from their skin. Then the highest MR studies we were able to see were between 2014 and 2023. So I would say AMR in wildlife has just become a new concern, and I'm glad for this workshop that we're having today, because it opens up the discussion. And what am I doing in this space? I'll say right now what I'm doing, I'm researching. I'm doing majority of my work is in AMR surveillance between livestock, human, livestock, and wildlife interfaces. So that's what I'm doing. I'm doing surveillance in humans, doing in livestock, and even in places where they coexist. That's majorly my work. And so what can we do now that AMR we've identified is a problem? One, what you can do, first of all, is antimicrobial stewardship. I don't know whether you've had this before, but it's one of the main agendas that are driving forward, that we only use antibiotics or antimicrobials when only it's necessary and in the right dosage, for example, and also restricting the use of critical antibiotics, especially in food animals, to preserve the effectiveness in human medicine, because we say there's one transmission event occurs between humans and livestock and to humans. So we want to cut this off by restricting at least critical antibiotics use. I think these are places we can go read more on. Another thing we can do is having a circular economy for pharmaceutical compounds by reducing their waste, reducing waste and pollution from pharmaceuticals, either by recycling, reusing these materials, or reprocessing and reusing antimicrobial agents, and reducing drug waste that is led into the environment. Another tricky one here has been waste treatment in innovation, having treatment systems that remove antibiotic residues. I think this would be a dream come true, but it's one avenue. I decided to present everything that we can do. And also treatment alternatives. I think these will be at the forefront of fighting AMR, and some of these are, for example, I'll talk about them later on, is the use of phages. Since the 1990s, we've barely come up with new compounds that target the bacteria in new ways. So we are running out of what we have. What we have, all we are doing is either progressing, advancing it, changing it, but barely are we coming up with new compounds. So bacteria are remaining to be the same. They're becoming more resistant. So the fight is really on, and treatment alternatives can really help before we come up with something new. So when it comes to wildlife, we realize standardized AMR testing and reporting methods have been very different in Africa. I don't know in the human sector. I've not been able to interact with other countries, but this is one major area, so that to be able to regionally compare notes and say this is what we're seeing. Let's do something about it. Also, collaborative surveillance, especially here in Kenya, is something we've been doing a lot. Having research that is cutting across the human-animal environment interface. Promising development. in the fight against AMR have been, first of all, the development of novel antimicrobials. But I can say very, very few have been developed. I think I've seen a very, very short list of new antimicrobials that have different target sites that the bacteria has not yet developed resistance to. Other prevention strategies may be like vaccines, passive immunization approaches, doing microbiome manipulations where you end up with healthy gut bacteria, for example, so that you can prevent infection. The game changer has been phage therapy, or we call them bacteriophages. Bacteriophages are this thing you have put up here, a picture that looks like a robot. This is a bacteria, and this looks like a robot that's trying to walk on top of a bacteria. But that's what they are. They are bacteria that are able to infect. They are viruses that are able to target and kill bacteria or infect bacteria and kill it. And the good thing about them is that they are very targeted. They do not kill anything else. Unlike when you take antibiotics, they kill the bad bacteria and the good bacteria as well. Phages tend to only go to the target bacteria that they are target for. They have an icosahedral head. These are like filaments, and they tend to have a long neck. So what happens when, for example, it's walking on top of the bacteria, this one gets contact gets contact with the membrane of the bacteria, injects through, pushes its material from the neck inside it. What it does, it gets into the bacteria and forces the bacteria to form its nuclear element. And it forms so many until the bacteria dies, and then it gets out and goes and finds the other bacteria. So its multiplication and action is how a bacteria like HIV affects human beings and their cells. So phages will get into a bacterial cell instead of a human cell. So they will only infect and kill specific bacteria, and they evolve alongside bacteria. So they are able to adapt to resistant strains, and unlike antibiotics that are the same all through, these ones they change, they become more powerful against the AMR. And of course, there have been success stories where phage therapy has been used to successfully treat drug-resistant infections, for example, Pseudomonas, Staphylococci, Klebsiella, and recent studies have shown that combining phages with antibiotics can actually restore antibiotics effectiveness against resistant bacteria. So it's still a new development. It's not been fully approved. It's still in experimental stages, but this is where we're going. I think that's the end of my presentation. I just wanted to tell us antibiotics should be a last resort drug. They are very good. They changed the way we saw the world of infections. They saved lives, but they should be a last resort drug. Not when you get a sore throat, you run to get an antibiotic. And please, let's finish our courses of antibiotics. Preach that. Let's save lives from AMR-related complications. Thank you.
Dr. Jemima, I should say I love the way you simplified antimicrobial resistance, and I hope all of us from here now we understand what it is and how it happens and what we can do individually and as a group. Thank you so much. Your presentation was very, very interesting. Yeah. So right now, before we start the Q&A, we'll jump to Muso, Professor Muso.
Thank you very much, Sarah. I hope I'm loud enough.
Yes,
Am I audible? Oh, thank you very much. Thank you. I need to thank Jemima. She has brought out very good technical aspects and she has simplified most of them. So I will go into my presentation also so that we can be able to share what we know about the AMR. Okay, thank you. I'll just remove the video. All right. So just like Sarah has outlined, we'll be looking at when medicines fail, how antibiotic resistance endangers wildlife. That's a very important aspect we need to look at. The first, I'll give you just an outline, a bit of an introduction to today's topic. Then we'll look at the global impact of AMR. Jemima has touched on that, and I'll also touch on that one in another angle, in another dimension. Then we'll look at the impact of AMR on the wildlife. Then, since I'm Zambian and I've done research in Zambia, I'm going to bring out a Zambian case study. I've got colleagues who have been working on the other pathogens, especially one indicator pathogen, which Jemima outlined, E. coli. But for me, I looked at zoonotic tuberculosis in wildlife because this has been a very serious issue. It's a borderline issue where we don't know if it is neglected or it is a disease which has completely been sidelined. So we need to see the case study which we have in Zambia, then the call to action. So in terms of introduction, antimicrobial resistance, this has emerged as a very complex issue. And currently, it is reaching a very dangerous tipping point. If you rank the global health threats, you will find that of all the global health threats, the top 10, you will find antimicrobial resistance in the top 10 global health threats. And just like Jemima has outlined, AMR, which is antimicrobial resistance, occurs when microorganisms, these can be bacteria, viruses, fungi, even parasites, they evolve and develop resistance to the available medicines, making it almost impossible to treat these infections. And because of that, it leads to high mortality, either directly due to antimicrobial resistant pathogens or indirectly. So the nature of this critical public health issue has got two primary components. The first is that the emergence of this AMR pathogen is quite varied, very diverse. It is a problem in livestock, it is a problem in wildlife, and it's even a very bigger problem in the human population. Secondly, the rate at which this AMR problem is spreading, the pathogens are spreading at a very alarming rate. And what we have seen is that the adaptation and survival of these dangerous pathogens are very well associated with the ongoing and the increasing natural selective pressure, which is anchoring or related to human, animal, and environmental health settings or interactions. Now, Jemima has talked about this issue. If you look at the deaths from drug-resistant infections, they are actually skyrocketing. And she outlined that AMR is set to surpass almost all diseases in terms of attributable mortalities by 2050. And you saw that she indicated that more than 10 million deaths will be occurring annually. And this is a very serious issue we are looking at. like I've said, it is reaching at an level becoming a serious global health emergency. And by 2019, WHO characterized it as such. So, like we have indicated through Jemima's presentation, whenever an antibiotic is used, there is also what is known as a selective pressure, where some of the bacteria escape that pressure and then proliferate or transfer their resistance genes. And some of these, when they escape, they find themselves either in the environment or they go either in hospitals as hospital infections, which end up infecting others. So over time, we've seen that bacteria develop what we call evolutionary mechanisms. They need to survive also to either external environments that they will defend themselves against other microorganisms, and they counteract the pressure of the body or the host's immune systems. But if you look critically in the human population and also the livestock population, the same scenario is true, where antibiotics are sometimes incorrectly used or even overused. So this misuse in animals or humans is also driving the AMR issue further and further. So the ability of the bacteria to contain these antibacterial resistance actually is moderated by 4 key factors. There is what we call the host-encoded specific factors. We are still under introduction. So I'd reached this stage where I was to explain how AMR reaches the environment. So the key issue with the AMR reaching the environment, if you look, there are 5 key aspects, and these were outlined a bit through Jemima. We have got the hospitals, then we've got the general population, then we've got also animal, especially intensive food animal productions. Then we've got aquaculture industry, apart from the industry itself, the general industry. If you look at the hospitals and the general population, there is what we call wastewater treatment plants. These, they generate a lot of antimicrobial resistant pathogens and also chemicals which build antimicrobial resistant pressure on the already existing normal pathogens. And these are released in the environment. It's the same with the intensive animal productions, aquaculture industries, and the general industry. This map, I was on this map. I wanted ourselves to familiarize with the map. You can see how the world looks like. One aspect, apart from Asia, Africa is the second largest continent in terms of the antimicrobial impact, with close to 4.1 million mortalities. So if we go a bit further, we look at the normalized global map in terms of disease burden, what you are going to discover is that the whole world shrinks a bit. You look at the Americas, North America, South America, you look at Africa, which is a bit in green, you discover that it bulges in terms of the disease burden, and also antimicrobial resistant are also in this disease burden. And that's where the biggest problem is. The models which have been formulated, they predict that this problem is going to actually triple or quadruple in Africa. Why is it so? The second map answers to this. Why? If you look at the availability of physicians, you look at Africa, it virtually disappears from the global map because the number of physicians available to attend to these global threats are very limited. You have got the highest disease burden, but the least capacity in terms of physicians available. And this is a serious risk factor. So the impact, if you look critically on the wildlife, one, wildlife, and I liked this because Jemima had outlined that it's very difficult to treat wildlife. Actually, if you come to countries like Zambia, under the national parks, once you enter a national park, you leave everything the way nature allows. You don't treat animals in the national park unless it is necessary. So wildlife becomes or acts as untreatable reservoir hosts for antimicrobial pathogens. And the resistant bacteria can also come out from herbivores and affect what we call predator-prey dynamics in the ecosystems. Jemima touched a bit on the birds, the migratory birds. We have also birds like the scavenger birds, which are super spreaders of these events because there will be any animal which dies, they will go and scavenge and spread the infections. But we've got also another danger, the migratory birds. We've got what we call the Eurasian African migratory birds, which migrate from Asia and Europe into Africa and back. So these, they are key in the dynamics of intercontinental spread. The AMR also has been seen to affect what we call the health of pollinator species, especially if you look at the bees and other key pollinator species, which will negatively impact even agricultural systems. The resistant microbes have also been seen to spread rapidly at the livestock and wildlife interface, and these have sometimes been seen to lead to either ecosystem collapse, especially in extreme cases, or to leading to serious threats in terms of classification of wildlife species under the IUCN. So, another aspect which is key in terms of wildlife is that apart from being the untreatable reservoir hosts, they can also be seen that this wildlife can actually be used in wildlife trafficking, the animals which are trafficked to other areas. There are also animals which can also be seen to cross-spread between the wildlife and the domestic animals, which can lead to increased mortality, not only to domestic animals, but to wildlife populations. Now, I want us to look at the Zambian case study using TB in wildlife. I think most of us are familiar with the African map. So I'll zero in on Africa. If you look, that is Zambia there. But Zambia is on the southern central part of Africa, surrounded, if you look at this map, by 8 surrounding countries. Tanzania, I know, is also, Tanzania is neighbor to Kenya. We've got Congo, DR, Angola, Namibia, Botswana, Zimbabwe, and Mozambique. These are all countries to South Africa, then Malawi. So we are very close. The ecosystem of Zambia represents a bit of ecosystem in the northern countries like Tanzania, near Kenya. On the southern parts, all these countries have got almost a similar ecosystem to Zambia. Now, if you look at the global incidence of TB, just by the correlation, the estimates of new TB cases of all forms, the 100,000 population, you look where Zambia is and the surrounding countries, close to more than or 300 per 100,000 population, new cases occurring. So this is a serious threat. Now, I've already indicated where Zambia is. Let's look at this case study. I want people to look critical on this. The first thing I'm going to do there is a national park and what we call the Kafue Flats. The Kafue Flats are key. Why are they key? One, they are home to a very unique, known as an iconic wildlife species, the Kafue antelope. The Kafue antelope is unique. Why is it unique? Because in this ecosystem, there is what we call a high intensity of livestock and wildlife interaction. So on top, that is the Kafue antelope. Then below you have got cattle. Then outside the Kafue flats, there are other ecosystems which keep cattle, which doesn't interact with the Kafue flats. So we had to survey in these. Apart from these, you see, the border towns here, why we looked at this, there is a Namibia, Botswana, and Zimbabwe around this, the interaction of these animals around these countries. Then there's another antelope up there, which is known as the black lechwe. It's found in the Bangweru Plains. Why did we look at it? We looked at this scenario because the Bangweru Plain in this area, the black lechwe, the sister to the Kafue lechwe, does not interact with any cattle. They don't interact with any livestock. Then we also looked at the red lechwe. Now, these are the pictures of the three iconic species of wildlife which are found in Zambia. They are actually endemic to Zambia, apart from the red lechwe. So, that is the Kafue lechwe antelope. It's a bit lighter in complexion, but very, very similar in height and all the other biometrics with the other two antelopes. Then B, that is the red lechwe antelope. Then C, that is the black lechwe, found in the northern part of Zambia, near Tanzania and near DRC, that side in Bangweulu. So these are the unique antelopes. Now I'm going to explain something with this. This is the Kafue lechwe. If you look critically, in the foreground, you are going to see the Kafue lechwe in the Kafue flats. In the background, you are going to see there are cattle there. That is the livestock in the background. And they interact in these ecosystems. And this is where the problem comes in, because what was earlier known was that the Kafue lechwe harbor bovine tuberculosis and they pass it to cattle. That was what the nation knew until when we went into to determine if this is indeed true, that the wildlife were the source of the TB into the cattle. So I'm just giving you some pictures here. We confirmed the TB in the Kafue lechwe antelopes, and we discovered that from gross postmortem, we were able to get about 4%. If you look on the right side, that is one of the lungs of one of the Kafue lechwe. Below is the heart. Serious tuberculosis, you even see it on the gross postmortem lesions. 17% microscope was positive, culture 27%, but we went beyond this to do even the DNA and also to sequence and to even look at the what we call the antimicrobial resistant patterns. So that was the approach which we used. You can see like on the A, that is in the lab, we used quite a number of media, LJ, Stonebrook, and also Midget to understand this. So once we did all this, we were able to even get very pure isolates, which we did advanced molecular techniques. This is just one of the publications which was done on tuberculosis in Kafue lechwe antelopes in preventive medicine. Now, let us go back to the Zambian map. the M. bovis strains. There were 3 key M. bovis strains. That's the TB strains, TB of animal origin. What we discovered, if you look, there was a strain under polygotyping. This is a spacer oligonucleotide typing technique, which we used. There's this strain internationally in the Microbacterium bovis, even in human TB. It's known as SB0120. We found it in Zambia. And it is, if you look under host, it's the only strain which was found also in the Kafue lechwe antelope. There are key genotypes. The other genotype was the SB0948, which was found, and SB0143. Let me give you this scenario. So, what did we postulate? So, most of the animals came with the trade routes, mostly through South Africa with the early settlers into the Kafue flats. So, as they move into the Kafue flats, you can see there was now with the Kafue lechwe antelope. And what happened is that there is a new strain which is closely related to SB120, which came from livestock. But what we discovered that both of these, SB120 and SB094, are resistant strains. But apart from this, and actually it is multi-drug resistant. By multi-drug, we discovered that this strain is actually resistant to the first-line drugs which are used for treatment of humans. I need to mention here that one of my colleagues who I worked with in our team was able to further isolate in humans from the medical side. He was able to isolate the SB012 strain from human in this same region where it was found in both cattle and in Kafue lechwe. There is also a paper which we did not too long ago where we were able to isolate the same drug-resistant strain in Chakma baboons. These are Papio usnus, and we published this in transboundary diseases. Then, as we move, there's another group of animals which had another strain known as SB0131. And this might have been spread in the cattle, mostly in the eastern part and outside the Kafue basin. This, we didn't just finish here. We went and did a further analysis. What we discovered that in these same areas, we looked at what we call the prevalence and the proportion. There were a lot of animals with this strain SB0120 in the Kafue flats and in no other parts of the country, just around this. And there were the same ones which were also found in the Kafue antelopes. Then we discovered that cattle had also other strains which were not in the antelopes. And this was the most resistant strain. So what we did was to even go now into the abattoirs, apart from to get all the positive cultures around the country. And as we did, we found that cattle actually indeed hosted a lot of these genotypes, but the 120 were found mainly in cattle coming from the Kafue basin, not any other areas. So what we discovered is that the isolation, there was another isolate which was closely related to the resistant one, but it also showed that it was drug resistant. So let us move to what is known as call to action. Based on what was presented by Jemima and what we've also seen in my presentation, there is what is known as call to action. The antimicrobial resistance multi-sector or multi-stakeholder partnership platform was launched globally in Rome simultaneously. That was in Rome, in Nairobi, Kenya, in Geneva, and in Paris on the 18th of November 2022. The thrust was to ensure that the growing threats and the impacts of AMR are addressed. Then there was the quadripartite, which involved the Food Agriculture Organization, UNEP, WHO, and the World Health Organization for Animal Health. They joined forces to underscore that the threat which AMR presents to humans, to animals, plants, ecosystems, and livelihoods needed a One Health approach, a combined approach to solve. And because of that, there was what was known as the One Health Joint Plan of Action. This is the magnitude. So the first Joint Plan of Action, 2022 to 2026, is actually very much in place and working together for the health of humans, animals, plants, and the environment, so that they can build capacity to respond to these issues. And one of the key issues is the issue of antimicrobial resistance. And the One Health approach seeks to improve the health of the entire environment, including humans, animals, and plants. So, one critical aspect about the quadripartite and the other organizations under the call to action is that we are trying to increase or strengthen global surveillance systems. I liked what Jemima outlined. If you look critically in Africa, she showed the paucity of data. There's very little data which speaks to AMR in wildlife. Very, very little. And the need to promote One Health approach cannot be emphasized. The issue of research in wildlife ecosystem related to AMR is very, very, very poor. The same with public awareness, waste management systems. How can we improve this? How can we train wildlife veterinarians in antimicrobial stewardship? How can we develop wildlife-specific AMR data for better tracking? I haven't seen countries, and this Jemima brought it also out. Do we have database? Because those with information, those with data, they'll be able to better understand, they'll be able to better track, and also make informed decision. So with that said, I want to request my colleagues on the platform to be able to ask questions. So I finished my presentation with just to show sometimes when you are working with wildlife, the kind of equipment you need. Thank you.
Thank you so much, Professor Musso. And it was very interesting to hear about the TB case study that happened in Zambia, to see that it is actually the livestock that were infecting the wildlife.
Absolutely.
Now I would like to open the floor for questions. Let's have a discussion with our two experts here. In the chat, there was a question from Fiona was asking which bacteria species are currently most resistant as we talk now.
That's a very good question. I'll answer it like that, like this. You see, if you look, it's not a very simple answer. Why am I going to say that? Because bacterial species resistance, it depends also on the quantum of resistance. But apart from the quantum, if you look critically at tuberculosis, it's the only bacteria actually which has reached what we call extensively drug-resistant pathogen. This is a pathogen which has gone, there is what we call drug resistance, DR, which is drug resistant, maybe to one drug. Then it goes to what we call multi-drug resistant, MDR. Then it goes to extensively drug resistant pathogen, Beyond extensive drug resistance, TB has reached what is known also as total drug resistance. So bovine tuberculosis, you might need to know that it is firstly very resistant to the first line of drug. One of them is pyrazinamide, and it quickly becomes resistant to rifampicin. Then, once rifampicin is resistant, you'll find that the two other drugs like ethambutol and isoniazid, they also become resistant. And this has been shown in bovine tuberculosis. But unfortunately, there is what is known as the second line treatment. And also, these have been seen to be also failing. There is a very recent paper which we did with my colleagues also from the human side. We were able to isolate from the same area where we found the SB0120. We found an SB120, which is actually now very resistant even to the second line TB drugs, and we published this also. So if you look critically, TB, I can say, is one of the. Why is it like that? Because TB is a disease which has got a bit of low virulence, but very high pathogenicity. It moves slowly, but the destructive nature of it is quite high. So that's the danger. Some other bacteria, why you don't see them having this kind of impact, it's because within 24 hours, their growth rate. But still, for tuberculosis, you have to wait for an average of 8 weeks for you to even culture it, to see the growth. 8 weeks. So it grows slowly, but it destroys the organs it affects. I think you have seen in the picture I showed you of the lungs, where it causes granulomas. It kills the lung capacity. So that's the danger of the pathogen we are dealing with. Yes. So, but it's not a very simple answer to just say it's tuberculosis, but the others, they've got their own forms. We've looked at other pathogens, but TB, it's a bit in its own class also. Yeah, because of its longevity. Thank you.
Thank you so much, Prof. Jemima, would you like to add anything on that?
I'd like to say thank you very much, Prof, for your elaborate answer. I've been able to read some of your papers, and it was a delight. I would say, like Prof has said, it's very the answer is very expansive because there are different classes of bacteria, the way they behave. So you're not able to really pinpoint and say bacteria A is the most resistant. No, so for every class, the way they present. But Professor has been able to bring out the issue of TB, which is what we are dealing with in Kenya. We are calling it zoonotic TB, and it's a menace in the human sector because of the characteristics they say there.
Thank you so much, Jemima. Yeah, I can see there is a hand from Chance Muntali. Chance, would you please unmute and ask your question?
Okay, thank you, Prof, for the nice presentation.
You're welcome.
Can you hear me? Yeah.
Yes.
So my question is, you've talked about tuberculosis in livestock and wildlife animals. So currently, for the two, treatment is not encouraged to treat in wildlife animals and in livestock. So my question is, does that encourage the mycobacterium to have like more time to mutate, or for them to mutate, they have to be exposed to bacterial medications first? Yeah.
Thank you, Muntali. Yes, that's a very good observation. The first thing like you have outlined, you see, TB is very insidious, and it's a disease which it takes time. You see, when your body immune mechanisms are okay, you can sequester it in the body and it doesn't even show. So sometimes it takes stress. When someone is stressed, even the animals, when they are stressed, nutritional stress, like we have seen in these wild animals, that's when you see the disease coming out. In cattle, mostly TB is diagnosed during abattoirs at postmortems. If it's not at postmortems, like in Zambia, we ask veterinarians to do what we call brucellosis and tuberculosis screening before you sell your milk, so that the milk is coming from brucellosis and tuberculosis free animals. What usually happens is that when you go and test, you'll find that this animal looks very nice, but it has got TB. And it has brought controversy where you tell the owner that this animal needs to be taken to the abattoir. You need to separate it because, like Mutari said, you cannot treat it in livestock. Why? Because of the economic implications. The same drugs, but unfortunately for animals, you cannot compound the drugs in form of like tablets, because these are tablets which humans get. But for animals, you need to compound them in injectable forms. And the treatment length is the same or even longer, because you might need to treat with multiple drugs for over 8 months. Now, you are treating an animal for over 8 months. One, just the economics don't allow. And also, the treatment outcomes are very unpredictable. So that is the main reason why you find that TB in animals is not recommended. For wildlife, it is even very difficult because these antelopes, you cannot, it won't even give you 50 meters. When you want to dart a wild animal, what is the success rate? The cost of, because in wildlife, we use what we call remote delivery. You need to have a dart, a syringe dart, which is quite expensive already. Now the drugs, and these syringes are a bit smaller for them to be able to be projected over a certain distance to dart an animal. Imagine you need a 20 ml volume of the drug. How are you going to deliver it? Most of these darts, they might be the bigger ones. If you find the 3 cc, which is the 3 milliliter darts, these are a bit on the bigger side. And beyond 3 to 5, they become even difficult. So for wildlife, it is just inhibitive just to do the process. For cattle, the economics don't allow, as well as the animal welfare issues. So these are some of the serious inhibiting factors which we don't use to, which prohibit us to proceed with treatment. And the other thing, like I've said, you only see this during either abattoirs or during a screening, like when you are doing tuberculization. Yeah, thank you. Unless maybe Jemima has another answer.
No, I don't have
Thank you so much.
a different answer.
Okay.
Thank you so much. And in the chat, we have another question from Simon. He's asking if illegal wildlife trade contributes to the spread of AMR, and what measures can be taken to reduce the risk?
I can go. Of course,
Yes.
illegal wildlife trade will contribute to AMR greatly because, for example, when we are I've been involved in a survey that was taking into account imported or trafficked parrots from Central Africa to Kenya. And what we were able to pick up is that First of all, when they're being transported, there's always this assumption that because of the stress that the animals will be under, medicating them with antibiotics is going to prevent either future infections or relieve stress along the treatment, the transport. So, one thing that these trafficked animals will do is that there will be that stress-induced infections that will lead to use of antibiotics, which we've seen will lead to development of AMR or at least its acceleration. Also, them, if they're harboring antimicrobial resistant genes from where they come from, their trade route or their travel route is already at a risk of being introduced to new ARGs or resistance genes that were not there. Environmental contamination along that route, of course, those are some of the key reasons why that we would say animals that are traded or are trafficked, even in the holding areas in the markets where they are held before they are transported, anywhere they stop along, you'll be able to think of it as a route where these resistant genes were able to traverse through, and they become hotspots for infection. Look at something like mpox. I know we're talking about AMR, but when you look at it in Kenya, you will see it has a route from being where it was imported. It will follow a consistent route. So those the trade routes that they'll pass through, those are hotspots for infection. What can we do about it? We can impose maybe stricter regulations for these. We can we can always regulate antibiotic use. We can improve hygiene in wildlife markets. We can have campaigns to improve awareness in these wildlife markets where they exist, or even input stricter measures in where they're not supposed to be. So yes, trafficking wildlife contributes to spread of AMR.
Thank you so much, Dr. Jemima. Anything to add, Prof?
Yeah, thank you, Dr. Jemima. Just like she has said, based on our experience here, I'll give two scenarios. One, if you look at the illegal wildlife trade, sometimes in terms of what we call game meat or wild meat, it comes from where there are illegal mass die-offs. You find that some animals have died without the actual cause being established. Some people get this meat, and sometimes the danger is that they actually adulterate it. They mix it with the livestock, cattle meat. I'll give you the scenario. We had the anthrax outbreaks. Some people are treating these animals with all sorts of antibiotics because they knew that there's this outbreak of anthrax, thinking they'll be able to immediately an animal, it was treated maybe in the morning, pumped with antibiotics, it dies. What do? And they are also wild animals, they would do mix. And people in towns, when they hear it's girl meat, it fetches a higher price. So the illegal trade, you can see the impact. So you find that people are exposed to these adulterated, because when you are getting illegal girl meat, you don't actually know exactly what you are getting. They smoke them together. There's a mixture of this livestock and these are the actual girl meat from these mass die-offs, and people are given this. So the food safety issue comes in. And like Jemima has outlined those factors, you combine with also the adulteration, the aspect of illegal wildlife spreading AMR is actually very, very high. And it's something which, if you look at also at the pathogens which are associated with illegal wild meat, they are quite high. There's a study which has been done and it's quite serious on that aspect. I submit.
Thank you so much, Prof. Is there any question from the audience?
I think I saw a question that was asking what was it? On phage therapy to treat AMR, what will be the byproduct of dead bacteria, and will that affect the host, either human or wildlife? I think I can answer that. So,
yes, please.
what phages are doing, it's similar to what an antibiotic would do. So it gets inside a bacteria, and a bacteria kills the bacteria. Of course, once a bacteria dies, it produces things we call endotoxins, polypeptides. There are things, peptidoglycans, that is. There are things that will come out of the bacteria that can actually harm the human being. And some of how it manifests, it's either you get sepsis and you get into shock. You also get what else do you get? You get a fever. So these are real scenarios, and they happen also when you're using antibiotics. So what we do, how we can maybe for and to say, if you're very healthy, if it's a healthy organism, either a human being, an animal, or wildlife, if they're already healthy, or they're at their livers and their kidneys are functioning optimally, the liver and the kidney are able to clear this off very quickly. These endotoxins, they clear everything very quickly. But if somebody now is totally immunocompromised and is being treated with phages, what we can do is dose them. Instead of, we don't give them at once. It's dosed similar to what you can do with antibiotics. So you have an extended treatment period to counteract the effect of the bacteria dying. This will be resolved in the body, similar to when antibiotics work. They become excrement, and they either pass through the liver and the kidneys into our excrement. And I think one of the most important things, or what I love about phages, is the ability to adapt, like a bacteria is adapting. They improve, they also build immune memory. Thank you.
And there is also another question from Simon. Simon, would you like to unmute and ask your question?
I'm asking, how do wildlife rehabilitation centers, like the snake farms or the zoos, how do they address the risk of AMR when treating injured or sick animals?
Orange.
Okay. Thank you, Simon. How do they address the risk of AMR when treating injured? So the critical aspect with AMR, if you looked at what Jemima presented earlier on, is to do what, if there is a need for antibiotics, to do an antibiotic susceptibility test. Before they treat with any antibiotic, which antibiotic is very effective, they need to be able to do. So you cannot do away with diagnosis. They diagnose and they do an antibiotic sensitivity test. Then they'll be able to treat effectively. And they need to follow the required treatment period. Why is it like that? Because there are also what we know as the pharmacodynamics and the pharmacokinetics of the drugs. So once they follow the treatment protocol, you find that you are going to avoid the risk of bringing AMR in these wild animals, especially like we have said in game ranches. Yeah, so that's my answer. Then I discovered that I didn't answer your other second part, what measures can you do in illegal wildlife trade? So some of the measures, you need what we call a multifaceted approach. Simple things like surveillance and monitoring. the issue of raising education, awareness, and also risk assessment by the departments of national parks so that they will be able to understand the risks which can occur due to illegal wildlife trade. So those aspects, once they're in place, you'll be able to avert or control the risk of AMR in illegal wildlife trade. Yes. Thank you. I hope I've managed to answer your questions.
Yes, thank you, thank you.
Thank you. Thank you, Prof. I wanted to talk to Simon. In Kenya, you find, because I've volunteered for the snake park before. What you find is unsubstantiated use of antimicrobials. For example, before then we used to have metronidazole, and you'd find pufferers, they come down with oral thrush or mouth rot of some sort. That was our first line of treatment without doing any anti-susceptibility studies. So, like Prof has said, sensitivity studies are very important. Sorry. Also, avoid overuse. Having practices that, let's say you're looking at a pufferer, you've rescued it from the wild, you're bringing it into captivity. That transition session should be important too, because we know these animals in captivity, they don't do very well, and sometimes they come down with infections that we rush to treat using antibiotics. So understanding their ecology, what they need, prevents illnesses, or what we can do is preventive medicine, prevent it as much as possible instead of just using them. For example, other snake farms in Kenya, because I visited a couple, we're using them as for tourist attraction. So client education is very important in this sector to be able to put this animal in its right, appropriate environment, prevent illnesses. And when it comes to treatment, let's do appropriate treatments. Consult veterinarians when you're treating. Let's not rush to take an antibiotics because we've seen diarrhea in chicken, so you think this drug it's meant for animals, it's for chicken, so you go ahead and give it to a snake. I think client education will be very important.
Okay.
Yeah.
And during this handling, is there a possibility of the handler or those dealing with wildlife, that is hand-on handling, can they also unknowingly transmit the drug resistance bacteria to the wildlife? Is there a possibility?
Yes. I would say yes, and I would say it very carefully, avoiding to implicate anybody. So what we say with AMR, transmission will, or what do you say? So we say something like transmission will infer direction. You can, there's something called colonization. So you have the bacteria inside you, you have the resistant gene. And by handling an animal, let's say you're handling an animal, you spend the day with this animal a long amount of time to be able to shed what is inside you and give it to this animal. It happens. It doesn't happen always. So in a population of 1,000, maybe one event can occur. So that's what we call the transmission event between either reverse between humans and animals or animals to humans. That happens, but we don't go holding our hands back because we are going to transmit AMR genes. It happens, but I would say I wouldn't. Yes, let me say it can happen. I don't know if Prof wants to expound more.
I think that is very sufficient. It's fine. Yes. Thank you.
Thank you so much. I can see we have one more questions in the chat from Chance. Chance is asking, can you please clarify on the overuse of antimicrobials? Is it the longevity or of treatment or the dosage which is acting as a factor in antimicrobial resistance?
Yes, actually, it is both. I say both. Why is it both? Because duration of treatment is key. Prolonged exposure, if you prolong the exposure to antimicrobials, there is what we call exerting what we call selective pressure. Because the more you treat beyond the required timeline, it is going to favor certain certain microbes which are going to survive. Those which are going to survive, they are the ones which are going to spread the resistant strains. And this was outlined in the diagram which Jemima had actually shared in the first few slides of her presentation. Then also dosage. If you look, under dosage contributes to MR because if you underdose, you are not going to the effectiveness or the lethal dose, what we call the lethal dose for the antibiotic to clear that infection doesn't reach the required level. So you remember when she showed us the bacteria cell with the efflux pumps where the bacteria it sucks in and like spits out the antibiotic. You need more at what we call a required lethal dose. But if you underdose, you will find that the bacteria, once it throws it out, there is no effective dose to destroy that bacteria. So it also leads to AMR. So both longevity and dosage can actually lead to AMR. There are other factors, but those two I can respond to accordingly, just like that. Unless if there's something which Jemima wants to put across.
No, there's nothing to add. I think you've brought it out very well. When we talk about antimicrobial use, misuse, we are talking about dosage and the duration, like Prof has very well explained.
Thank you so much. And to finish, we have a question from Andrew, which says, are we really using the right antimicrobials? Least effective medication, or we need more studies?
That's a very nice question. I don't know if I have to start preempting. There's another country because you see one aspect about antimicrobials, like Andrew has indicated, is also the purity. There are some also what we call counterfeits. You find that you need an antibiotic written, for example, 200 milligram, maybe sulfonamide. But when you go to test in the laboratory, you find that instead of the 200 milligram, you find an antibiotic with actually maybe 80 milligram. Yet it's supposed to be 200 milligram. And the doctors and the pharmacists, when they prescribe, they have faith that what you are getting 400 milligram, but you are getting 80 milligram. Already, that is a disaster. So counterfeit is another, just now, Jemima was talking about underdosage. Even counterfeit is a form of underdosage. And like Andrew has said, these become least effective. And like he has said, we need more studies to really, that's where a number of countries have got the standard bureaus to look at what are our people taking? Do they have the right medicines? If a pharmaceutical company brings an antibiotic, let's say penicillin, and they say this is 400 milligram penicillin, but you find that the active ingredient, instead of being 400 milligrams, there's only 60 milligram. That is already an underdose. So that's a very good question. Are we really using the right antimicrobials? We need the standard bureaus to check, even the pharmaceutical regulatory agencies in the countries. Thank you.
Thank you so much. I can see Andrew. Andrew is raising his hand, but then we are running out of time. Andrew, I want you to go back home very happy, so you can unmute and just make your comment.
Thank you. Thank you so much. In fact, I'm so impressed with the professor's response, and I commend on Professor Musso and Dr. Jemima's presentation. These are very insightful presentations. And in fact, I'm so concerned. We scientists may be contributing to all these problems that we are causing. It happened. I had a issue of bacterial infection, and I attended one hospital. They gave me a medication which seemed to be like an overdose. But when I applied that medication, I didn't get any remit. So I had to go to the other hospital. And they told me that's the worst treatment you got. They had to give me another medication. You see, now the body is being over pumped with the medication. And this is the problem. We may say it's a microbial resistance, but sometimes we scientists are causing this resistance because of either underdosing or overdosing. So thank you so much. I think this is this should not be the first discussion. I think we need to discuss further. I see this challenge here in Tanzania. Here in Serengeti National Park, we see these challenges. I see some studies, people are coming up with some studies on the resistance. But also, I was also thinking, is it an interrelation with the useful bacteria, or are these harmful bacteria linking or affecting even the harmful bacteria to the useful bacteria to evolve and become harmful again to the environment? I was trying to think, Professor and Dr. Jemima, maybe if you can help me understand further. I know we are running short of time, probably. It may not be possible now, or I can share my contact, then we can discuss further on how we can improve this. Thank you so much, our host, Stella Malaga, for giving me this opportunity.
Thank you very much. So, very good observation from Andrew. The presentation by Jemima, she also indicated that if bacteria, different species, both harmful and useful bacteria, are sharing the environment, and then, let's say the harmful bacteria, they've developed resistant genes. There is also horizontal and vertical transmission. They are able to transmit those genes even in the beneficial bacteria. So that is the danger. Imagine in the stomach, there is this harmful bacteria, which is releasing all the resistant genes. And these resistant genes can actually also be transferred to a beneficial bacteria. And that is the danger. You are very right, Andrew. But like we have said, it's a very big topic, which you can share your contacts. We can still flow seller. We can still discuss it out of this platform, unless there's something which Jemima can say. Given the time factor, I think I'll end there. Thank you.
Thank you so much, Prof, and thank you, Andrew, for your questions and even your narration. I think that's a problem that cuts across everywhere. It's a problem where I am right now. I took a personal survey one time, and I had a sore throat, but I was offered azithromycin. we've been able to, or literature even tells us, like, for example, if you have pneumonia from, let's say, Klebsiella pneumoniae, azithromycin will not work. But you come to our Kenyan hospitals, that is the first line of treatment that you're given. You go to pharmacy outlets, the first line of treatment for almost everything is always an antibiotic. But it's a war to fight. It's a broader problem that we need to keep discussing, keep talking about it. And I think the message will hit home. And also always, if you can, we have the movies to have evidence-based prescribing. Like Prof has said, let's do antimicrobial resistance tests, sensitivity tests. Let's see what do you have, what antibiotic actually works for what you have, and then now you can be given that. Underdosing, I think Prof has explained it very well. It's a problem we're having in this country, and I'm glad I sit next to two colleagues who are carrying out the research in the country, collecting antimicrobials from from livestock, majorly poultry farmers, who are using more, we've discovered are using more antimicrobials in the country, but we are doing that. So it's research that is ongoing. We've also asked ourselves that question. Are we using the right antimicrobials? Are we using them at the right dosages? What are we getting from the pharmacies that is licensed? Is it the right quantity? So we do random surveillance, we collect them, and if you go to our organization's page, you are able to see what research has been done, preliminary summary reports. I think it's a topic that let's preach it, let's talk about it more, and it reaches to the grassroots so that our parents back in the village, for example, where I come from, they'll know that not every time they go to the chemist or the pharmacy or the hospital, they need to come back with a dose of antibiotics.
Thank you so much. Thank you so much, Andrew, for bringing in a very important discussion. Yeah, it's about 9 p.m. So we should end the discussion here. And I hope from this discussion, you've understood what antimicrobial resistance is and the effects of antimicrobial resistance to our ecosystem. And lastly, what we can do, what part we can play in preventing antimicrobial resistance. I'm wishing you a good night, and thank you so much to our experts. Thank you so much for your time. We really appreciate your time, and have a good night.